with, and they're controlling basically everything that the nervous system has to do. So if we zoom in to even deeper level, all of those proteins are encoded by our genomes. We each have 23 pairs of chromosomes. We get one from mom, one from dad, and on these chromosomes are roughly 25,000 genes. They're encoded in the DNA, and the nature of a given cell driving its underlying biochemistry is dictated by which of these 25,000 genes are turned on and at what level they're turned on. And so our project is seeking to look at this readout, understanding which of these 25,000 genes is turned on. So in order to undertake such a project, we obviously need brains. So we sent our lab technician out. We were seeking normal human brains. What we actually start with is a medical examiner's office. This is a place where the dead are brought in. We are seeking normal human brains. There's a lot of criteria by which we're selecting these brains. We want to make sure that we have normal human between the ages of 20 to 60. They died a somewhat natural death with no injury to the brain, no history of psychiatric disease, no drugs on board. We do a toxicology workup. And we're very careful about the brains that we do take. We're also selecting for brains in which we can get the tissue, we can get consent to take the tissue within 24 hours of time of death. Because what we're trying to measure, the RNA, which is the readout from our genes, is very labile, and so we have to move very quickly. One side note on the collection of brains, because of the way that we collect and because we're requiring consent, we actually have a lot more male brains than female brains. Males are much more likely to die accidental death in the prime of their life, and men are much more likely to have their significant other spouse give consent than the other way around. So the first thing that we do at the site of collection is we collect what's called an MR. This is magnetic resonance imaging, MRI. It's a standard template by which we're going to hang the rest of this data. So we collect this MR, and you can think of this as our satellite view for our map. The next thing we do is we collect what's called the diffuse and tensor imaging. This maps the large cabling in the brain. And again, you can think of this as almost mapping our interstate highways, if you will. The brain is removed from the skull, and then it's sliced into 1 centimeter slices. And those are frozen solid, and they're shipped to Seattle. And in Seattle, we take these, this is a whole human hemisphere, and we put them into what's basically a glorified meat slicer. There's a blade here that's going to cut across the section of the tissue and transfer it to a microscope slide. We're going to then apply one of those stains to it, and we scan it. And then what we get is our first mapping. So this is where our experts come in and they make basic anatomic assignments. You could consider this state boundaries, if you will, those pretty broad outlines. From this, we're able to then fragment that brain into further pieces, which then we can put on a smaller cryostat. And this is just showing this here. This is frozen tissue, and it's being cut. This is 20 microns thin, so this is about a baby hair's width. And remember, it's frozen, and so you can see here old-fashioned technology of a paintbrush being applied. We'd take a microscope slide, and we very carefully melt onto this slide. This will then go into a robot that's going to apply one of those stains to it. Okay. And our anatomists are going to go in and take a deeper look at this. So again, this is what they can see under the microscope.